So India is blessed with 5 types of silkworms. Starting with Mulberry silkworm, that contributes a major share, about 70% of the total silk production in India. So Mulberry silkworm basically is produced from Bombyx mori. It belongs to the family Bombycidae, and the rest of the 4 silkworms, which are shown here, they are regarded as one-year silkworms. So here in one-year silkworm, the major ones are like tropical Tasar, which is produced by Antheria mylata, oak Tasar by Antheria prolei, and eri silkworm by Samia cynthia resini, and Muga by Antheria assamensis. Here, whatever I am delivering today, they are not only just for the like knowledge enriching, they are also very much useful for competitive exams like JRF, SRF, NET, or ARS, or any other competitive exams related to entomology. So I request all the participants to kindly take a note on whatever I'm saying here. So you can because you can't get a comprehensive material over Tasar sericulture in any book. So these are all latest innovations that are happening in the Tasar sericulture. So because now in the exams they are asking the current updates. So I request the participants to take a note on that. So coming to the tropical Tasar, so here the major important things you can see here in the figure. So here the unique feature of the tropical Tasar is that the peduncle is very hard. So the peduncle is, if you see in the oak Tasar, the peduncle is very weak. So you can see in the figure, the peduncle is hard in tropical Tasar only. And you don't find any peduncle in eri silkworm, which is produced by Samia cynthia resini. And in Muga also the peduncle is very weak. If so far there was no such type of questions asked in the competitive exams, but we can expect such type of questions. So peduncle is very, we can say that hard in case of tropical Tasar. And another unique feature is that in Muga silkworm, it's ending to Assam region. So that's why it is called as Antheria assamensis. Eri silkworm is called as Ahimsa silk. Why? Because here the filaments are not continuous. Here the silk is not reelable, so it has to be spun. So the filaments are discontinuous. So there is no need of killing there is no need of stifling here. So there is no need of killing the pupa. So straight away we can spun the silk here. So that's why eri silk is also called as Ahimsa silk. So sorry. Coming to the unique features of Tasar silk industry. So here we are straight away Tasar silk industry. We are straight away we are dealing with the tribal development. So here rural employment we are directly dealing. We are providing the opportunity for their as a livelihood aspect. And other thing is that India is blessed with a lot of forest cover, and this forest cover we are utilizing for growing of the Tasar silkworms, as well as this industry provides, just a minute. Okay, this industry provides livelihood to more than 3.5 lakh people. So and also this culture is not a culture, it is more than, it's like a heritage. So many tribals, as a part of their culture, they practice the Tasar sericulture. And also Tasar industry provides employment opportunity to the rural women in the form of reeling, weaving, fabric making, etc. And also this industry does not require like a high investment. It is like low investments, and the farmers are extracting high returns from this industry. And also the fabric of the Tasar silk is unique from that of Mulberry silkworm. Coming to the core focus of Tasar industry is that, so here if you see many of the forest areas in Odisha, Jharkhand, Chhattisgarh and some parts of West Bengal, where migration is another issue. So farmers or rural people, they are migrating to the cities. So because they are unable to harvest the livelihood activities in their rural areas, so they are migrating. So we are with our latest technologies, we are encouraging the farmers to take Tasar sericulture, so that reduction in the rural migration can happen. And also rural women empowerment is another aspect which we are trying from our institute, and also poverty alleviation and self-employment, because here if you see a lot of school dropouts, college dropouts, we can see in the rural areas. So we are trying to motivate them so that they take up Tasar sericulture as a self-employment opportunity. And also we can bring the school these dropouts into the sericulture, so that they don't go to the extremist ideology which happens in the forest areas, where tribals, they get influenced by the antisocial elements. In today's presentation, I will be dealing with how Tasar sericulture is practiced in the practiced by the farmers, how they are selling, what are the cost-benefit ratios, and other latest innovations which are happening in the Tasar sericulture. So that I am going to talk in today's presentation. So starting from the seed to the fabric, so all the activities we are going to I'm going to present in a nutshell form. Basically, Tasar sericulture, I will be dealing tropical Tasar only, that is Antheria myelata. So that is basically that is grown on Arjun, Assam and Sal plantations. So here you can see, here these are the block plantations. These are block plantations. These plantations are maintained by the state government or central government offices, where we maintain it 6 feet or 7 feet height. And we manage them. We take up pruning, pollarding activities, so that the height is maintained, and we grow the silkworms over them. And after growing silkworms, the cocoons are harvested, and they are hung in the grainage houses in the form of garlands. You can see the garlands here, and from these garlands, the moths emerge. So rural employment is happening at each and every stage, starting from the plantation to the fabric. There are a huge scope for rural employment. Say, for example, plantations. Plantations in Arjun, Assam plantations, the farmers are making nurseries and they are selling at the rate of 10 rupees to 15 rupees per sapling. And after rearing of Tasar silkworm, farmers, they are selling the cocoons at the rate of 3 to 6 rupees per cocoon in the open market. After the spling and the silk can be extracted, and raw silk cost 1 kg of raw silk is somewhere around 4,000 to 5,000 rupees. And later the fabric can also be sold in the form of saris, dupattas, shawls, jackets, like that. At each stage, Tasar sericulture is providing huge opportunity for the rural employment and tribal development. If you see, in a nutshell, I have given some of the statistics here. There are ample opportunities. If you see seed production, in seed production, one DFL, one disease-free laying is 60s to 70s. There was an extensive survey across the country, and we have recorded, our institute has recorded somewhere around 44 eco-races, and they are distributed starting from Kashmir to across the country till Kerala. There are a lot of eco races there were recorded in way back 1960s and 70s. And India, as I said earlier, India is blessed with a lot of forest cover, mainly dominated by Shorea robusta, that is 86.9%. And later, the next comes is Arjuna and Asan. Plantations are abundant in India. So these are the primary host plants for the Tasar silkworm. So in exam, if somebody asks what is a primary host plant for Tasar, means somebody should you should able to answer that Arjun, Asan and Sal are the primary host plant. And most of the wild ecoraces, say for example in Odisha, Modal ecorace is present in Simlipal biosphere reserves in Baripada, those areas. So if you see these wild ecoraces, they are mostly found in the forests of Sal. So coming to the list of the primary host plants, that is Terminalia tomentosa Asan, Terminalia Arjuna, that is Arjun, Shorea robusta, Lagestromia, Parviflora, Speciosa, Indica, Ber, even Gigifus, Mauritiana, and Hardwickia binata. These are some of the primary food plants for tropical Tasar silkworm. For temperate Tasar silkworm, it feeds, that is Antheria prolei, that feeds on Quercus serrata, that is oak. It feeds on the oak tree. Coming to the secondary host plants, we are having some secondary host plants like Terminalia chebula, Bellarica, Catappa, Paniculata, and Jamun and Shorea tellura. So these are some of the secondary food plants for tropical Tasar silkworm. If you see, as I said earlier, we are having 44 ecoraces starting from Kashmir to here Kerala. So here mostly Jharkhand is known as Tasar capital of India. So this one should note, Tasar capital of India is Jharkhand, and even we can see some ecoraces in Odisha, Chhattisgarh, Madhya Pradesh, and some northeastern areas, and in UP and in Andhra also we are having one ecorace. In Karnataka, one ecorace is there, and in Kerala, one we have found one ecorace that feeds on cashew. So these, so we can say that Tasar silkworm ecoraces are spread from Kashmir to Kerala. Coming to the variability. So see here, Tasar silkworm, it is having huge variability. You can see the cocoon size, shape, as well as the peduncle length. It varies. It varies from within the host plant. If I rear a ecorace in Jharkhand and Odisha, the cocoon characters they change because environment plays a huge influence over the cocoon characteristics. Not only the environment and also the food, the type of food crop on which we are growing. So there exists a huge diversity in the morphology of the ecoraces. So at present we are even studying how many ecoraces at present we are having. That for that we are running we are having a project where we are going for a huge survey. So the present status of the ecoraces in India. Coming to the most important commercially exploited Tasar ecoraces. So at least we should remember some of the names like Daba, Raili, Modal, Sukinda, Laria, Sarihan, Bhandara, Andhra local, Japla. So these are some of the commercially exploited Tasar silkworm ecoraces. So these ecoraces are named based on their locality, tribal people. These names are mostly given by the tribal people and based on their locality. So that's why if you say for example Modal is found in Odisha, Raili in Chhattisgarh, Daba in Jharkhand, Andhra in Andhra local, Bhandara in Maharashtra. Bhandara is a place, and in Andhra it is present in Warangal areas. Sarihan, it is also present in Jharkhand and Bihar. Laria is also found in Jharkhand. So these are the some of the commercially exploited Tasar silkworm ecoraces in India. My slides are moving, right?
Yes, sir.
Okay. Now, so far we have seen the ecoraces, the commercially exploited ecoraces we have seen. So now we'll start the rearing. So before rearing, we take up the disinfection as a activities. So disinfect, because Tasar insect, the immunity of the insect is very weak. So once disease comes, means we can't save the silkworm. So that's why we take all prophylactic and preventive measures so that the silkworm, we harvest a good crop. So we take up the disinfection activities seriously. So there is a schedule from CSRTI RANCHI. We are recommending to the farmers, and farmers take up those activities like spraying, dusting, mostly bleaching and lime. They will be dusting, and some insecticides if required, and also some fungicides. Coming to the rearing, here rearing, we are rearing the silkworms on the host plants in order to harvest the cocoons. So here the phases like chalky rearing and late age rearing. So why in detail I am telling is that in ARS also, so far they asked in ARS or any exam, they asked only mulberry. Mostly they are asking mulberry. So there is a high chance that in near future they may ask about the Tasar silkworm rearing also, Tasar and Muga. So that's why in a simplified manner I am trying to explain here. Coming to the chalky rearing. Chalky rearing means engage rearing is known as chalky rearing. Here we will be rearing the silkworm up to 2 stages. That is first instar and second instar. We are rearing the silkworms on host plants which are maintained at 4x4 spacing. Say for example, in a spacing of 40 into 30 by 10 feet. So it can accommodate 70 plants where we can easily grow 250 DFLs. So one DFL, it contains 200 eggs or 2 grams weight of the eggs. So we generally weigh the Tasar eggs and we distribute to the farmer in the form of DFL. If suppose a packet contains 100 grams of egg, then simply we can say that it contains 50 DFLs. So one DFL cost is 12 rupees. So here we do the chalky rearing under the nylon nets, because the first and second instar, they are very fragile in the sense they are attacked by the birds, ants. So that's why we rear the first and second instars under the nylon nets, under the protected conditions. So later, third, fourth, and fifth instars, they are reared on the economic plantations, which are maintained at 6x6 or 10x6 spacing, and their height is 5 to 6 feet. Coming to the life cycle of the Tasar silkworm, here you can see in the first image where male and female moth, they emerge from the cocoons and they mate immediately. So here sexual dimorphic characters exist, and please note the sexual dimorphic characters. Like male moth is having bipectinate antenna, their abdomen are slender and even thinner, I can say narrower when compared to the female, and their wings are also dark, shorter when compared to the female, and female abdomen is filled with eggs, and his antenna is I can say like weakly developed bipectinate, I can say it is not fully developed, and after mating immediately female start laying the eggs. The eggs hatch. The egg period is 8 days. So means after laying the eggs, on the 8th day, the eggs start hatching. So here you can see the first instar coming out of the eggs. Okay, so there are 5 instars in Tasar silkworm. You can see the second instar, third, fourth, and fifth instars, and you can also see the cocoons with the peduncle that are hanging from the branches. Okay, and the peculiar behavior of the Tasar silkworm is that immediately after hatching, it will eat its own eggshell as a source of nutrition. And here you can see in this photograph, you can see after molting, it is a second instar, and you can see the molt skin also here. And in this photograph, you can see the spinning of the Tasar silkworm by taking the shelter of the leaves. As I said earlier, that we generally give DFLs on the weight basis. So this packet contains 100 DFLs. 100 DFLs means it contains 200 grams eggs, and the cost of 1 DFL is 12 rupees. And you can see in this here the life cycle, again the same thing. The eggs get hatched, and we rear them on the Arjun Asan leaves, chalky rearing under the nylon nets, and later we'll shift them to the economic plantations, which are maintained at 6 feet height. And if you see the weight of one larva, it is somewhere around 40 grams. You can see the fifth instar will about to spin. If you take measure the weight, they are somewhere around 35 to 40 grams. And in this photograph, you can see how it is spinning the cocoon. As all animals suffer from diseases, Tasar silkworm also suffers from diseases. So here, some of the peculiar diseases which you have to note, typical symptoms you can see here. This is pebrine. So now pebrine is no more a protozoa. It is under the kingdom fungi. You should keep a note on this. Here, black pepper-like spots that are found on the silkworm, then we can say straight away that is infected with the pebrine. But there are more than 14 types of Nosema miletensis. Nosema miletensis that we have reported somewhere around 14 strains of the protozoa that are infecting the Tasar silkworm. And sometimes we don't find even the pepper-like spots also, but still then they cause the infection. At the adult stage, the moth will be crumpled with the crumpled wings or scaleless abdomen. So these are also some of the indications that the silkworm that the moth is infected with pebrine. So such type of moths, they are not used for the egg production. So we'll collect them and we destroy them. Coming to the second disease, that is viral disease, virosis, that is caused by CPV, a cytoplasmic polyhedral virus. So here typical treetop symptoms we can observe. And here also, if we find any diseased worms, we collect them and we destroy them because they will act as a secondary source of contamination. So these are some of the polyhedral bodies under the SEM. And again, these polyhedral bodies, they survive in the field for 6 to 10 years. So that's why here we take the disinfection in the field very strictly. Coming to the third disease, that is bacterial. So here a group of bacteria, they cause bacterial diseases in same as in Muga and as well as in the Tasar, the same. So here you can see the typical symptoms here, like chain type of excreta. So here excreta will be in the form of chains. And coming to that rectal protrusion, its rectum get protruded outside. And third symptom is anal lip sealing. The silkworm anus gets sealed and the litter will be blocked there. And fourth is bacterial flutchery. So here there is a combination of some virus as well as bacteria. They cause the infection. Hello. Good afternoon. मैं थोड़ा बिजी हूँ, थोड़ी देर में बात करेंगे. Sorry. So these are the four symptoms that we can find in the Tasar silkworm. That is chain type of excreta, rectal protrusion, anal lip sealing, and bacterial flaturing. Coming to the fourth disease, that is muscardine, that is caused by fungus Penicillium. So here what happens is that there will be a full mummification of the silkworm, and you can see the mycelium that spread over the silkworm, and the silkworm becomes hard, and if we break the silkworm, it will give the sound, popping sound, just like a chalk piece. So if you see, now we have completed four diseases. That is, one is pebrine, caused by Nosema miletensis. Second is virosis, caused by CPV. Third is bacterial disease, caused by a group of bacteria. And fourth is a fungi, that is fungal infection, muscardine disease. And coming to the major parasites and predators of the Tasar silkworm, like we are having two endoparasitoids like Blepharipa jebina, Uji fly. So this scientific name is very important. So Uji fly of Bombyx mori is different, and Uji fly of Tasar is different. So here Blepharipa jebina is a dipteran. So that is an endoparasitoid, and another ichneumon fly. So we read hymenopterans are beneficial, but here the one of the hymenopteran, that is ichneumon fly, Xanthopimpla predator, is a pest. Coming to some of the predators like stink bug, redovid bugs, wasps, certain ants, praying mantis. These are general. These two wasps, red ants, and praying mantis. These are generalized one. Here, Canthicona and Sicanus, they are like nuisance, or they take a huge toll. If you don't manage properly, they take up to 30 to 40% of the loss. So here, Uji fly, if you see Blepharipa jebina, it lays egg on Tasar silkworm, which is about to spin the cocoon. And these eggs, they hatch and they enter inside the body, and they complete their life cycle. And when they want to emerge, they make a hole on the cocoon on the bottom side. So this is the key characteristic. If we find a cocoon with a hole, small hole on the downside of the peduncle, the opposite side of the peduncle, here the peduncle will be there. If the hole is on opposite side, then we can say that the silkworm is infested by Uji fly. The same case is ichneumon fly. The ichneumon fly also, it emerges from the top side. If you find any hole, small hole on the upper side of the cocoon, that is near to the peduncle, then we can say that it is infested with ichneumon fly. So these two important characters, please remember. So downside hole, it is Uji fly. Top side of hole, it is ichneumon fly. Coming to the stink bugs, these are predators. Generally, up to third instar, they are a nuisance and they take a huge toll. And you can see these bugs, they are sucking the hemolymph from the silkworm, and you can see that the silkworm dies because of the this stink bug. And coming to the redovid bugs, these redovid bugs also, they suck the hemolymph there, and they kill the silkworms. These are the praying mantis. You are well aware. And some of the wasps like Vespa orientalis, they carry the first and second instars, and they kill them. So now we'll see the grainage activities. So now we have read the silkworms successfully. Say for example, we have read, and now the cocoons are about to harvest. So we have harvested the cocoons. on an average, if I go for a 1 DFL rearing, 1 DFL rearing means it contains 200 eggs. So even after the losses by diseases, pest, environmental conditions, whatever, if we also on an average, a farmer can harvest 40 to 50 cocoons easily. So one cocoon cost is somewhere around 4 rupees, 3 to 4 rupees. Even if we keep at 3 rupees also, so 50 cocoons, 3 rupees means it is almost all 150 rupees. Means farmer is spending 12 rupees and within 45 days he is getting 150 rupees by selling from the cocoons. Okay, so these cocoons, after harvesting, farmer, he will segregate, he will cut the branches because the peduncle is attached to the branch. So with the help of secateurs, he will cut the branches and he will remove the cocoons with the intact peduncle. See, you can see the cocoons with the peduncle and also ring will also be present. And he will if we have to go for the seed production, then we will, what we do, we hang them in the form of garlands. So you can see the garlands tied to the bamboos. So grainage here, the important thing is, what is grainage? Grainage is a place where disease-free layings are produced. So DFLs are produced in grainage. Grainage is a facility. See here the cocoons are like garland. So we make garlands and we hang them in the grainages. So here the another important thing is that in a garland, 5 cocoons we make it as a single bunch. So such type of bunches, 20 bunches are there in single garland. So in a single garland there are 100 cocoons. So just for a convenient, we made like this. So one garland contains 100 cocoons with 20 bunches, and each bunch contains 5 cocoons. And we keep a proportion that, like in a bunch there will be 40 female and 60 male. So in the we just try to maintain that proportion. And the bottom, from the floor of the grainage, we generally keep 60 centimeters above the garlands will be hung 60 centimeters from the floor, and also between the garlands we give 15 to 20 centimeters spacing, and between the rows also we make like, see, you can see this one row of garlanding, and this is one row. In between these two we maintain 45 to 60 centimeters for the proper aeration, circulation, for inspections, for ease of moving. For all these things we maintain, we keep a spacing of 45 to 60 centimeters. So inside the grainage room, temperature and humidity, they are constantly maintained, like 30 to 35 degrees centigrade, and humidity 40 to 60%. So we maintain that. So these are some of these structures like grainage houses, like Pakka grainage house, mudwall grainage house, tubular grainage house, and green shed grainage houses. So these are some of the photographs of the grainages. See here, generally the mating happens immediately after emergence. So in this photograph, you can clearly see this is a male moth and this is a female moth. Okay, so here we can see the eye spots. So these eye spots are to protect the, it is like a behavior, a morphological character that moth is having to avoid the predation. So the birds will feel that these are eyes and they attack these eye spots so that the abdomen and the important organs, they get safe. They won't get attacked by the birds. So this is a behavior, this is morphological protection, we can say. After mating, what we do, we just cut 1/3 of the wing, just as a marker, as an indicator that this particular moth got mated. Okay, just for a mark. After mating, we will decouple them and we will cut the 1/3 of the wing, so that we'll come to know that this moth has already been mated. After mating, we'll cut the wings, like 2/3 of the wings we just cut and we keep in the earthen cups, and we'll keep for 3 days. So till 3 days, whatever eggs it lays, so we'll just go for mother moth examination. If mother moth is free from the diseases, then we take all the eggs. If mother moth contains pebrine, then pebrine spores if we find, then whatever eggs it laid, so we just discard them. So here, unlike in Mulberry, so here in Tasar, we go for individual mother moth examination. In Mulberry, suppose there is a 1000 worms are there. There we take 1 or 2 moths, means there we go by lot basis. But here we go moth basis, single moth basis. Okay. So these are some of the egg laying devices. Like this is an earthen cup, this is a paper cup, and plastic boxes are also used for egg laying. Coming to the mother moth examination. One thing everybody has to remember that in entire sericulture, mother moth examination is done only to detect the pebrine. That's it. We don't check any other diseases. So that's why here in a nutshell how mother moth examination is done. So after egg laying in the box for 3 days, so we will take the mother moth, we'll cut the abdomen in the mortar. With the help of pestle, we'll crush the mother moth. Before that, we'll add K2CO3, which digests the causticize the tissue, disintegrate the tissue, and liberate the spores. And we'll crush with the pestle, and we'll take 1 drop of. Generally, we can go with centrifugation also, and without centrifugation also we can observe. So this protocol where centrifuge is available, there we go for centrifuge at 4000. With cover slip, and we'll observe under the microscope. If you find any spore like this, you can see here spores just like rice grains. It will be visible like rice grain with Brownian movement, lightly eggs this moth has laid. We'll just simply discard them. We'll destroy them, including the paper, including this paper box. Okay. Recently, we have, because you have seen this entire process, this is a traditional process where we have to go K2CO3 addition, centrifugation, and again adding the KOH. There is a multiple steps are involved. So to avoid this, we have developed one technology that is called pebrine visualizing solution that was developed from my section. So this simplifies the process, and in this photograph you can see the spores. You can see the spores. You don't find any other tissues. Okay, only simply spores you can visible. You can see. So the speciality of this solution is that if you add a single drop of this solution, it will disintegrate entire tissues, fat bodies, other silkworm tissues, and it will liberate the spores. And in the field, you can observe only the spores, if at all present. Okay, so that is the beauty of this solution, and this solution is being commercialized, and it works excellently in Mulberry, Muga, and Aries also. Now in entire India, this solution is being used for mother moth examination. So now the eggs have passed the mother moth examination. So what now we are doing here? Now we will collect all the eggs. So here we will collect all the eggs. We'll scrape them. We'll collect all the eggs. So you can see the eggs just with all you can see they are dirty with a lot of unwanted materials. Wing materials are there. So now we have to wash them, because on the surface also there will be pathogens. So we have to wash them. So we wash them. Now we have developed a solution that is called Dupratex, wherein in a single go it can disinfect the surface of the eggs, and it also clean the eggs. So see here that the eggs are cleaned. All the artifacts are removed, and surface has been sterilized. Because why we have to sterilize the surface? I already said that in the earlier that silkworm is having the habit of eating its own eggshell. If on the eggshell, if suppose a pathogen is present, so silkworm will feed the eggshell, and while feeding it will take the pathogen also. So that's why we have to sterilize the surface of the egg. So for that I said Dupratex is again developed from my section, where it is a disinfectant. It is a surface disinfectant, cleaning as well as disinfectant in a single go. So this the eggs, if you see in this pouch, the eggs are collected in this pouch. Dip in the 5% of the Dupratex solution, just 5 minutes, and later you just start washing. If you finally we can see the eggs which are creamy color with eggshell. We can see. Okay. So later we will dry the eggs in the this dryers. So this is a box where in the bottom there are blowers are there. So they will pump the air upwards and will spread the eggs, and within 15 minutes eggs get dried, and we will weigh them and we'll make the packets like this. So here 3 things we will mention in the eggs. That is the name of the eco-race, that is here TV. TV means a trivoltine. So DABA TV, and also the date of coupling. So suppose date of coupling is on 4th. So on 4th it got coupled. You can just see date of hatching is 12. So within on the 8th day they will hatch. So date of coupling, date of hatching will mention, and the quantity. So this contain 100 DFLs. So 100 DFLs means this packet contains 200 grams eggs. Okay, so this is all about seed production. So this is all about now. This is up to here. It is all about seed production. So suppose some cocoons, they are diverted for the silk purpose. So here, what happens? The cocoons they are brought to the post harvesting centers, where they boil the cocoons. Here the cocoons are very hard. Unlike mulberry cocoons, we can boil them in hot water and we can just start reeling. But here we have to use hydrogen peroxide, strong alkalis we have to use to soften the cocoons. So once they get softened, we will start reeling them. So there are different machines our institute has developed. So by using these machines, the silk is reeled. So these are some of the machines like Kamadhenu, MRTM. This is improved MRTM, and finally we'll get the yarn. So these are some of the silk reeling machines. So once the silk has been reeled, you can see the Tasar silk has been reeled, and later we go with the fabric making, and that weaving, we'll go for the weaving and other like coloring, dyeing. So there are a lot of processes there. So these are some of the fabrics that are made out of the Tasar silkworm. See here, the fabric is unique. You can say, unlike mulberry, it is very unique. So it is having a what is a separate look. Even the saris are also they are having elegant look, superior quality, and they fetch the huge revenue to India through the imports. So far we have seen the traditional way. So far we have seen that the farmers are rearing the silkworm only for the fabric. Okay, they are selling the rearing the silkworms, they are harvesting the cocoons, and they are selling for the purpose of the raw silk production. Now we'll see, now it's the era of byproducts. So now from here we'll see what are the types of byproducts that are present in the Tasar sericulture, which can help to alleviate the poverty of the tribal people. Is my slides are moving, right?
Yes, sir.
Okay. In India, total silk production, if you see in 1920, here if you see the mulberry stands at the first position, second is the eri, Third is the Tasar. So here around 3,100 metric tons of the raw silk we have produced in 2019 and 20. If you see the unique of the Tasar silkworm, if you see the cocoon weight it is somewhere around 10 to 15 grams, and shell weight it is 1.3 to 3 grams. It is uncomparable with any other silkworms. If you see, so that is the one of the added advantage for Tasar silkworm, where it generates a huge amount of byproducts, which we'll see, which we have quantified, and I will be discussing in the coming slides. See, Tasar byproducts, after reeling, we just simply throw the pupa. Okay, I mentioned that one pupa weighs somewhere around 10 to 15 grams. We are just weighting that much weight of the protein as a waste. Okay, and the litter, silkworm, it generates huge quantity of litter. Fiber waste. This fiber waste in post cocoon, for getting the reelable silk, we just weigh from one cocoon somewhere around 0.2 grams of the fiber waste we are generating. Okay, from one cocoon, single cocoon, 0.2 grams of the fiber waste we are generating, as well as sericine. As I said earlier, the Tasar cocoon is very hard. We have to use alkalis to soften the cocoon. In this process, the sericine is a protein, that is a soluble protein that is present over the filament, so that get dissolved. So we just simply throw that sericine. Okay. Another is after mating, male and female moths, we just throw them. So these are the major areas where we can look for the byproduct utilizations. So I will be dealing in the coming slides what are the different types of projects we are taking, what are the technologies that are in a pipeline to reach the field. From a single cocoon, 0.3 to 0.5 ml of the cocoonage, it is generated. 0.3 to 0.5 ml. This cocoonage, what it is cocoonage, I will be dealing later. Just remember, so the cocoonage is 0.3 to 0.5 ml, litter 60 to 70 grams per larvae, and chitin 50 micrograms. Okay, 50 mg, 50 milligrams per moth, and fiber waste, I said 0.2 to 0.3 grams per cocoon, and pupae 1.5 to 2 grams. It is a dry weight basis. Okay. On dry weight, it is 1.5 to 2 grams, and again sericine is 100 to 150 mg per cocoon. So these are the if you see the quantity on from single cocoon basis. What are the uses of these byproducts at industry level? If you see, per year we are generating sericine. We are just simply throwing sericine, 280 to 300 metric tons of sericine. Okay, which is having industrial application like cosmetics, anti-cancer, biomedical, textile industry. Likewise, cocoonage also, it is having industrial applications. And its scale of production is 7.5 metric tons, and pupae 6,200 metric tons, and 4 lakh metric tons of the litter we are just simply throwing, which can act as a source for as a substrate media for growing the microbial culture at the industry level. Okay, and also fiber waste 800 to 900 metric tons. Okay. So these are the, if you say the byproduct status in India, so these are the latest statistics, I can say. So like I said, silkworm, from silkworm we are generating huge waste. Likewise, the trees, before rearing and after rearing, we go with the pruning, pollarding we do, and we generate a lot of waste. Either from the sal, arjun, asan, or oak, we are generating every year a lot of biomass. Okay, this biomass we simply, tribals, they are using as a firewood, and which in turn causes the indoor pollution and affects the tribal women. Because of the toxic gas is being affected. Even government of India is promoting to switch over to the gas. What is that? LPG. Okay, so and we don't know statistically, we don't have any data, so how much tree waste we are generating. So right now I am working on a project where we are trying to convert this huge biomass into some wealth by growing some medicinal mushrooms. These are some of the medicinal mushrooms which love the wood. These are some of the wood-loving medicinal mushrooms like maitake, lion's mane, turkey tail, shiitake, and some of the orange bracket fungi and Ganoderma. So these are some of the wood-loving fungi, and they grow well on the this wood. For example, Ganoderma, it is known as immortality mushroom, and the price of 1 kg Ganoderma on a dried basis is 300 to 600 rupees. Okay. Shiitake is somewhere around 1200 to 600 rupees. And biological efficiency means here, if you see the biological efficiency means if I take 1 kg of 1 kg of the wood. If I take 1 kg of sawdust or wood, okay, I just chop it and make it as a sawdust and make it as a packet. If I grow mushrooms over them, I can get 40 to 60 percent. Means from 1 kg I can get 400 to 600 grams. Like that, I can take 3 to 4 crops. Means from 1 kg, so around 1200 to 1400, I can get 1400 grams of the mushroom I can get. And if you just imagine in terms of monitoring, so if I grow Ganoderma from 1 kg of the wood, I can get 600 rupees or 800 rupees I can get, okay, by selling that mushroom instead of using that 1 kg wood as a fire for the as a firewood. Okay, so even after harvest of these mushrooms, we can use that substrate as a manure. After sterilization, we can use it as a manure or animal feed. Okay, so by this way, instead of using the tree waste for the fire, which lead to the toxic gases emissions, we are putting into use so that it helps the tribal empowerment, especially for the women. So here, steps in the Ganoderma production, if you see, first the wood, maybe branches, maybe dried leaves, whatever it may be, the wood we are tree biomass we are converting into the sawdust. This sawdust, after adding suitable amendments, they are made into the packets, 1 kg packets. We will sterilize them. Later, after the sterilization, we will add the Ganoderma inoculations, and we will maintain the optimum conditions under the culture rooms, and we'll get these Ganoderma fruiting bodies. We will harvest them, and we'll go for 2 to 3 croppings. So, okay, you can see the Ganoderma fruiting bodies here, and we'll harvest them, we'll dry them, and we can sell them. So this is the mass production protocol. You can see here the mass production protocol of the Ganoderma and the shiitake are more or less similar. Only temperature conditions differ. Okay. Coming to the second byproduct is sal seed oil. In India, sal seed availability is somewhere around 15 lakh tons. All these statistics we have taken from the State Department website. Okay, so this is the potentiality, that is 15 lakh ton. Okay, what is the potential of sal fat availability? Sal fat is nothing but if I take the sal seed, if I dry them, roast it, and if I extract the oil at room temperature, it will be in the form of fat. Okay, so if that sal fat is somewhere around 1.8 lakh tons. But at present, we are harvesting only 6,000 to 9,000 tons only. Okay, so there is a huge scope. Okay, who are the key players here? Again, Jharkhand, Odisha, Chhattisgarh, and Madhya Pradesh. They are the key states where sal seeds are used to extract the oil. Okay, farmers, say for example from Odisha, farmers, they are collecting the sal seed and they are selling at the rate of 20 rupees per kg. Okay. And you can see the fatty acid composition of the sal seeds also. You can I have listed here. This I have taken from this paper, where this Delhi Technological University they used sal seed oil for the production of biodiesel. Okay. What are the uses of this sal seed oil? So sal seed oil is used for cooking, soap making, as a substitute in the chocolate manufacturing, as a substitute for the cocoa butter, Okay, and also as a biodiesel. Okay, and also some people they use to adulterate ghee also, for adulterating the ghee. Another from the salt tree, another byproduct, we can say that another product that we can harvest is that resins. So these resins, also called as salt dammer or Bengal dammer, that is used for making the agarbattis, incense sticks, which are used in the religious ceremonies. Okay, or some inferior quality paints, varnishes, and also some to treat the diarrhea and dysentery. In the local medicine, sal resin is being used. Even Government of India has started a scheme, that a mechanism for marketing of the minor forest product. So they have given a minimum support price also. So as I said earlier, that the what is the rate? That is 2,000 per quintal. So per quintal it is 2,000 rupees, and 1 kg is somewhere around 20 rupees. And here sal seeds are directly the farmers, the tribals, they collect from the forest and they sell in the open market. Now due to COVID, many tribals they have affected, and they are demanding to increase the this price MSP from 2,000 to 5,000 rupees per quintal. So in a nutshell, how biodiesel is produced from the sal seeds? See, sal seeds, these are the seeds with wings. So these seeds, they are collected by the tribals. They dry them, and we can extract the butter. This is at the room temperature. It is called as sal butter or fat. If we simply boil that butter, it will be transformed to the sal oil. After esterification and washing and other processes, we will finally get the biodiesel. As I said earlier, the same group from Delhi Technological University, they have checked the emission characteristics and performance of the sal biodiesel, and they found that at high load, even the sal seed oil, it is emitting less smoke, and also it is even the engine performance is far superior than the traditional diesel. So here we can say that sal oil is having huge potential for the biodiesel in the future. Coming to the next component, that is sericin. Sericin, I should say that in the if you see the silk filament, in the silk filament, the core one is called as fibroin, and the outer covering one is sericin. So these two are proteinaceous material. This Tasar silk contains both sericin and fibroin, and during our cooking process, the sericin is being dissolved in the cooking water, and we are simply throwing that. Okay, so here a project that was run by Dr. Karambir Jenasar. So he is the expert on sericin, and he has standardized the protocol how to extract the sericin from the wastewater. So that I can say 1 gram of pure sericin is somewhere around 14,000 rupees. 1 gram of pure sericin. So if you recollect how much metric tons of sericin we are producing annually, you just calculate how much worth of rupees we are simply unable to utilize them. And here I have given the differences between the fibroin and sericin of both Mulberry as well as Tasar. So Tasar sericin is unique in the sense it is not like Mulberry silk. Okay, here both the fibroin and sericin, they are very much different from the Mulberry. So like I said, every year we are just simply throwing 143 crores of the business potential, and this project, the main aim of that is to how to extract the sericin from the wastewater. So the sericin, it is having the biomedical applications in I can say in the wound healing, preparation of lenses, drug delivery, antimicrobial activities, etc. So coming to the protocol here, how they are doing here. So the sericin, it is dissolved in the water. Say for example, after cooking, after cooking, this is the wastewater we are simply throwing as of today. Okay. This cooking water, they will add the some solution they will add, that is a patentable one. So they here we will add a solution. So after adding the solution, what happens? Whatever the soap, soda we are using, they get sediment here, and we'll get a supernatant that contains the soluble sericin. After this soluble sericin, we'll go for the filtration, and after a series of filtering, so we will get the pure sericin. And if you see, you can see the some images of the pure sericin. Coming to the fibroin, as I said earlier, from one cocoon we are generating 0.3 grams of fiber. So this fiber waste we can which contains fibroin. Okay, fiber waste contains sericin as well as fibroin, but we can go with the fibroin. So which is again having the biomedical applications like skin regeneration, making the bandages, membranes, drug deliveries. Okay, so even bone regeneration, like so biomedical applications are there. So again, this institute, again Dr. Jena sir, he has developed a protocol to digest this fiber waste and into in a solution form. After that solution is converted to powder as well as gel, and we can preserve that. So he has successfully developed this protocol, which can be used in the biomedical. So like I said that again this hydrogel is used to as a bandages, and a mega project in collaboration with BIT is under preparation. Coming to the another byproduct, that is cocoonage. What is cocoonage? Cocoonage is a proteolytic enzyme. After you have seen the cocoon. Inside the cocoon, pupa is there. If adult moth has to emerge from the cocoon, it has to produce some enzyme to soften the cocoon. Okay, it has to make a hole and it has to come outside. So that how that hole is made? Because of the with the help of the proteolytic enzyme that is produced by the moth. So this moth produces the cocoonage. This cocoonage dissolves the shell, and moth can easily come outside. So as of today, to soften the cocoons, we are using soaps, soda, hydrogen peroxide, and many strong alkalis, which is having deteriorating effect on the fiber. Okay. So now under this project, it's a DBT funded project where we can able to isolate the pure cocoonage and its variant, and we have developed what is a protocol for extraction of the cocoonage enzyme, as well as its variants, which can be used as a alternative to soften the cocoons, to entire cocoon. Say for example, you can see here while emerging, the moth releases the cocoonage enzyme. See, it has softened here. So here we can able to extract the variants, extract the variants which is having the same effect, just like cocoonage, natural cocoonage, that is being used as an alternative to for the soaps, so that we can go for the organic silk production. See here, it's a controlled one and it's a cocoonage treated one. And here also you can see in the photograph, this is the impact. You can see this is a cocoonage, it is a normal treated one with soaps and soda, where you see the natural color has been lost. And this is a cocoonage treated one. Here in these images also you can see this is a cocoonage treated one and this is a soap treated one. This cocoonage treated fabric, the silk, raw silk, it is having it has maintained its original color. Okay, instead of the soap. So coming to the pupa. So silkworm pupa, after reeling, we are just throwing the silkworm pupa, which is not being used for any purpose. They are simply degrading in the soil. So now we are having suitable technologies where we can use the silkworm pupa as a poultry feed, fish feed, in the cosmetics, in the medicinal application, human consumptions, and as well as making the compost. And we'll talk one by one how silkworm pupa is being used by Central Tasar Research and Training Institute, Ranchi. So here, silkworm contains, like I said, silkworm it contains a lot of bioma. Silkworm is just like a protein material. So here in the silkworm, it is a complete protein and it contains all required essential as well as non-essential amino acids that are required for the human beings. And it is why it is called as a complete protein, because it is in line with the FAO WHO standards. See here, in 2018 and 19, somewhere around 6,000 metric tons of the pupae they are generated in India. So this now we are making suitable technologies where this pupa can be converted as a fish feed, poultry feed, animal feed, etc. Coming to the profile, the pupa, silkworm, Tasar silkworm pupa, it contains protein 50 to 60%, carbohydrate 3 to 4%, fat 20 to 21%, and fiber 4%. So generally we remove this fat. So that is a important point here. So here we don't we defat the pupa first, and later we use it to make it as a feed. And also it contains vitamins and minerals. So here I have given the list. And this defatted silkworm pupa protein, it is white in color and it doesn't have that inherent odor, and it does not absorb moisture, and it is a food grade animal protein, and it contains more than 75% of protein and less than 2% of the fat. Coming to the next byproduct, so that is Tasar's waste to wealth by Cordyceps that currently I am running. So here I said earlier that a lot of pupa, 6,000 metric tons of pupa, we are just simply throwing. So this, sorry, and also per year, last in 2018-19, we have Tasar sector has achieved 46 lakhs DFL production. So for 1 lakh DFLs, we are using on an average 4 moths are required. Okay, so after egg laying, we are simply throwing the moths. Okay, male moth or female moth tissue, we are throwing them. We are throwing pupa, we are throwing silk moth, as well as if suppose any diseased moth is there, the eggs that are laid by that moth also we are throwing. Means here egg, larva, egg, pupa and adults we are simply throwing. Okay. Now this project aim is to grow a medicinal mushroom, which is a costly mushroom that is having huge medical application, that is Cordyceps fungi. We are growing on the Tasar silkworm waste material. So coming to the what is Cordyceps? Cordyceps is a fungi. Cordyceps that is having huge medicinal application. And it is a first reported entomopathogenic fungi, belongs to the family Ascomycotina. This is Cordyceps sinensis. This is also called as Asugamba, and many traditional Asian countries, they use it for the curing of many diseases. Okay, it grows on the Himalayan ghost moth. Okay, this is the larva. So it gets infected with the this fungi, and during the spring season, the fruiting body comes out of the soil in the Himalayan mountain ranges. Okay, it is harvested by the Chinese, Nepalese, Indian people. They climb the Himalayas and they collect these fruiting bodies, and they sell in the international market for 16 lakhs to 20 lakh per kg. So you can see in these photographs, you can see how the Tibetan, you can say Indians or Chinese, they climb the Himalayas during the spring season when snow melts. They climb the mountains and in search of Cordyceps. And here you can see they have collected the fruiting bodies. they clean them and they sell in the international market. You can see these are the cleaned ones. And here you can see the life cycle of the fruiting body stages of the Cordyceps. And because of the indiscriminate climbing, indiscriminate harvest of this fungi in the Himalayas, Government of India has kept this ban on the climbing, and also it is listed as red list under the vulnerable category. Okay. So this is my book chapter that was published in Springer, where we have given the full details about the Cordyceps and its medicinal properties. In a nutshell, Cordyceps, they are having a lot of medicinal properties, like it improves the immunity, it cures many major diseases including cancer, it is having cosmetic value, it enhances the stamina for the sports persons, and also it increases the fertility rate, and also it reduces the stress. Lot of medicinal applications are there. And here the main component is cordycepin. So this is the cordycepin is the main active ingredient which helps, which makes this fungi in demand. So in India, we import lot of cordyceps from China. Okay, because we don't have that much production in India. So that's why we depend on the China and other Asian countries. So that's why my project is again a CSV project, where we are converting the Tasar waste to wealth by growing Cordyceps. So we got the Cordyceps cultures like militaries, Cordyceps militaries from different regions of the world, like Thailand, Korea, Japan, and also some ICAR strains also we bought. And these strains are performing well, and we can able to harvest the Cordyceps and inside the lab. So here with the cultures, coming to the Cordyceps mass production protocols, here in 14 days, the culture we can grow on the potato dextrose agar plates, and the conditions required are 14 to 15 days and 20 degrees centigrade, and we can able to grow the pure cultures. And next is that broth preparation. Broth, how we are going to spawn production, how we are going to prepare the spawn. Here a bit of pure culture is dropped in this broth and kept in 4 days in shaking, and then we get the spawn. And after that we prepare the in these bottles we prepare the silkworm waste material, whether it may be pupa waste, adult waste, or egg waste. We'll make it a substrate and we give the inoculation, and we maintain the sufficient conditions like temperature and humidity. We maintain them. In 55 days we can get the fruiting bodies. You can see the fruiting bodies, and after 55 days we harvest them, we dry them, and we pack them, which can be used for the as a after harvest we can use these fruiting bodies straight away. We can consume that like 1 gram per kg, 1 gram per day we can consume that, or else we can also store them and we can sell it to the in the international market or a domestic market. Here after harvesting the substrate, I can say that substrate, that also we dry them, we store them. So this substrate again we can use it as a poultry feed or fish feed, which again improves their reproductive as well as other parameters. Okay, so that's thus our project is not going to generate any waste. So we are using the waste and we are not generating any waste. So this is a zero waste generating project. So here you can see the even by using the waste pupa also we can able to grow the Cordyceps. See here, these are all Tasar pupa inoculated with Cordyceps. As I said, this is a zero waste generating enterprise, where here straight away these fruiting bodies that can be dried and that can be used. So this is one of the brand in India, available in India, that is called Sanjeevani Chai, where they are also selling the Cordyceps. But in India, the point is that these people are growing the Cordyceps on cereal based. They are growing on rice, majorly on rice. But entirely our project is that we are growing on the silkworm waste material. So that is a novelty of our project. So from the substrate, this substrate after drying, that can be used straight away, that can be used as a fish feed, or else we can extract the pure cordycepin that can be used in the making of the shampoos, soaps, because it is having anti-aging properties. Coming to the next byproduct, that is chitin. So here the chitin, you know that insect exoskeleton that contains chitin, and the soluble form is chitosan. Okay, chitin is insoluble. So here you can see that this is Tasar pupa chitosan. Again, our institute has developed a protocol to isolate for the mass level isolation of the chitosan from the pupa. And this chitosan, it is having industrial application like healing of wounds and drug delivery systems, and also some antibacterial properties. Again, silkworm pupa oil. So here the silkworm pupa oil, it is having again an industrial application, like it is having linoleic fatty acids. So a lot of fatty acids are there that are having industrial applications. What are the applications of the pupa oil? Like alpha linolenic acid that can be used in the medicine as a food, high-end cosmetics. I just want to say, when I visited Japan in 2019, I have seen many of the Japanese cosmetics. Japanese cosmetics are high-end cosmetics. They all contain one or the other silkworm, this one material, whether it may be fibroin, maybe sericin, maybe like alpha linoleic acid. Okay, so all high-end cosmetics, they contain these things. So many Asian countries, they use silkworm not just for the silk, they use for these high-end industrial applications. Even in the coming slides, I will show another example. So this is one example. So in India, it is again a big problem. So we don't generally take it silkworms as a food, but Asian countries, they take silkworm as the food. But in future, maybe there will be chances or opportunities to convert silkworm pupa as a food source. So as of now, we are not that much interested to take silk insect as a food. And again, many Asian countries, they make wine, food items, chocolates using the silkworm. And these are some of the crafts again. So this one, tribal people in Jharkhand, and we give training to the Jharkhand in the Jharkhand, so that they make some of the value-added products like keychains, flower vases, souvenir items. So that can be prepared and that can fetch higher revenue to the tribals in the fairs, international fairs and national fairs. As I said, this is another example. So we have visited this lab that is in the University of Tokyo, where they are using silkworms. They are using mulberry silkworm. Okay, so but they are using silkworm to test the anti-diabetic drugs. Means they are using silkworm as a laboratory model organism. Okay, likewise in Tasar silkworm, whole genome is available. So we completed the whole genome sequencing, and there is a lot of opportunity in future also. Any basic research, we can use Tasar silkworm as a model insect to understand the, to screen the drugs or to understand the evolution. Okay, for basic studies we can use the Tasar silkworm. So as I said, Tasar silkworm litter. So here we are producing somewhere around 900 to 800 to 900 metric tons of litter every year. Okay, so that can act as an excellent source for the mass production of BT, compost preparation, extraction of the chlorophyll components that are used for that again has an industrial application. So here I have found that even silkworm litter is being used in making of the pillows, which is good for insomnia and arthritis treatment. Okay, in the biogas production, organic fertilizer making, and also extracting some chlorophyllin and trichothanol compounds. Now we have seen many byproducts, and also there are some of the challenges, like we don't have suitable startups, like we don't have sufficient startups in India, and also many project findings doesn't reach the field. Say for example, any university does any good work, but finally it won't reach the field. Okay, lack of public-private partnerships, and also lack of cost-effective feasible technologies, and market related issues. Say for example, my Cordyceps. So here the first question arises is that where to sell, how to sell, like that. But nothing to worry, we are also trying that in another 1 year. So this mass production protocol, either sericin, fibroin, Grainage, and also Cordyceps and Ganoderma, Shiitake, these technologies will reach the field, and we are mainly aiming the school dropouts, college dropouts, and some enthusiastic tribal people. So we train them so that they take up the entrepreneurship. Okay. Another major is that social stigma. As I said, so far, as of now, we are not taking silkworm as a food. But in some northeast regions, they take eri silkworm as a food. Finally, I conclude with some key points here. That Central Silk Board is emphasizing more on byproducts. Now, byproduct is this era is of byproducts, waste utilization. So I said, mass production protocol of Cordyceps, sericin extraction from the wastewater, and Grainage for the organic silk production, pupa as a fish feed, poultry feed are in pipeline to reach the field. Okay, and there is a huge demand that in coming years, tribal people are going to utilize these technologies for their upliftment, self-employment, and also it helps to realize the potentiality of the Tasar industry by the tribals of this country. And moreover, I would like to emphasize that as of now, the Tasar silkworm is viewed only for the silk purpose. So in the coming years, they will use byproducts, which is going to enormously change the lifestyle of the stakeholders. And so far, I try to emphasize many points related to the Tasar sericulture, and which are very much useful for your competitive exams as well as for academic purpose also. If any more information you require regarding these entomology preparation for SRF, JF, or NET, or ARS, you just contact me. Thank you.
Thank you, sir. Indeed, it was a very informative presentation. Sir, there are some doubts from the participants. Can I ask?
Sure.
Yes, sir. Sir, Bhupen Kumar Shahu has asked that as the Tasar cocoons are collected from the wild conditions, they must be exposed to the unfavorable environmental conditions. So is there any provision to prevent such damages?
we have not, the silkworm, Tasar silkworm was evolved in the field, wild conditions. Okay, naturally the cocoon is very tough, and it can protect itself from the natural vagaries, whether it may be temperature, rain, humidity, pathogens, whatever it may be. It is having sufficient accommodations to overcome these abiotic stresses.
Yes, sir. Thank you, sir. Sir, can I go to the second question?
Sure. You can.
Sir, next question is, as Tasar cocoon is cocoon marketing is tribal based, it must be exploited by the middleman. So what are the steps government has taken to prevent such exploitation and pay the farmers their desirable prices for cocoons?
Yes, actually everywhere the middleman, they exploit the farmers. Okay, so for that we made a we are having a minimum support price, that is 3 rupees, flat 3 rupees. Even in the field, there is a huge demand, even at 4 rupees, 5 rupees, even sometime at 6 rupees, the cocoon prices they go. So generally state government departments, they try hard to safeguard the measures, and as I said, minimum support price is there, 3 rupees per cocoon.
Yes. So the next question is, like, what are the government schemes that are implemented for the upliftment of wellness agriculture?
See, there are a lot of schemes are there, like Subplan is there, Mahila Kisan Sasaktikaran Pariyojana is there. So these are the schemes, central government schemes, which upliftment like Tasar industry in India. MKSP, Tribal Subplan, TSP, now Silk Samagra.
Yes, sir. Sir, next question is, like, is there any horticultural crops to rear this worm?
Horticultural crops, like I said, berry is there, cashew is there. These are the two crops where you can take up the silk industry.
Sir, Kiranbhai Pradhan, he has asked, like, you have mentioned 1 DFL as 200 eggs, but in case of mulberry silkworm, it is 1 DFL equal to 400 eggs. So on the basis of what components, like the number of eggs per DFL is fixed?
See, what is say in mulberry, 1 DFL is 400 eggs. But here 1 DFL is for suitability, 2 grams is 1 DFL. So it contains roughly 180 to 200 eggs. Okay. So that is for the feasibility only we kept. Here the eggs are given in the form of loose, like individual in the form of loose. Mulberry, they are given in the form of sheets. Okay. Where the eggs are they are stick with the help of glue on a paper. So there is no that such type of system, only as based on the feasibility only we are given.
Yes, sir. So it's really insightful session, sir. I must say, like, all the participants we are here, our participants are from different state universities, central universities, and there are many research scholars also. We are quite happy, sir. We are getting participants from many states and many, we have some international participants too, sir. So I must say all of them will be getting a very insightful knowledge from your like store of knowledge, sir. So, and we are very thankful to you, sir. We are getting some of your valuable time. We know that we are very busy being a scientist. So we are very thankful to get a time of your valuable time. So on behalf of our Faculty of Agriculture, Sri University, we want to thank you, sir. We want to convey our hearty thanks for your valuable time, which you have diverted for all of us, for enlightening all of our knowledge. And secondly, I want to convey a very hearty thanks to our Dean Faculty of Agriculture, Sri University, for motivating us to conduct such sessions. And I want to convey the thanks to all my organizing team for this like uniting and conducting such program. And lastly, I want to thank all the participants for showing their interest in this session And yes, all of them must say that it's a very insightful session. Thank you so much, sir. And we are hoping for such more sessions if possible, sir. We know you are a very busy person. If some valuable time we can get in for the time, we are hoping for that too, sir. Thank you so much, sir. Thank you all the participants present over here.